All right, thank you, Michael, for inviting me. Thanks for the pleasant introduction. I work with Esteban at UCSF, and I'm going to tell you today about how, I guess the theme is how precision medicine has changed your life. Well, it certainly has changed my life, and I'm going to tell you about professionally how it's done so. Forward. So some disclosures. I've founded a couple companies, advised for some others, number of grants and from industry and academia. So this is a precision medicine talk. I don't really have to go through this slide, but one of the first times I heard about precision medicine in the public was the State of the Union 2015. I'm sure Esteban had something to do with this. Because before we call it precision medicine, we call it personalized medicine, meaning, and I'm going to tell you a story. When I was in medical school, when I finished my PhD was on neuropsychiatric disease and how we use genetics to personalize medicine for autism and ADHD. So this is a big data talk, and the big data for this part of the talk comes from the Center for Applied Genomics at CHOP, the Children's Hospital of Philadelphia. So for those who may or may not know, before, one of the most major pediatric genomic centers in academia is CHOP, top pediatric hospital in the world. Something like 1 in 10 rule-outs go through CHOP, like in America. It is absurd. And since 2006, 2007, they've been building up this database, much like what Esteban has been doing, sort of with Gala and Sage. They've been doing it at this huge catchment of at least 1 million patients a year. Patients come in, they approach them at phlebotomy, they ask the parents for blood, and they ask, most importantly, if they could reconsent. If they consent them for recontact, meaning can we contact you about your child if we find something with this study you're enrolling in longitudinally. And when people think about genetics, they think about sequence, and this is a different kind of genetics. This is a difference in copy number of genetics, meaning I'm looking at duplications and deletions in genes. You can imagine there's 3 billion odd base pairs in the genome. It's really hard to tack down any one base pair and the functional significance of any one change in base pair. But if the whole exon of a gene is duplicated or deleted, I mean, that's a pretty significant, you would expect a pretty significant effect. So trisomy 21 is a great example of a duplication having a significant effect, a genetic duplication. But we are using the same, at least for this part of the talk, we're using the same technology that is used for sequence analysis. So 23andMe, Ancestry.com, it's all based on genome-wide SNPs, or single nucleotide polymorphisms. And technically, we don't need to get into the details, but it's an optical process, meaning how does this work? They plate samples on this chip looking like this. It goes into a machine, and the machine shines light onto the sample of DNA. Depending on which probe binds to that sample, the light differentially fluoresces. So it's essentially an optical process. And this is the raw data that comes off these machines. And based on this raw intensity, they make calls of allele 1 or allele 2. Except I'm not concerned with the alleles. I want to use the actual raw intensity to measure how much genomic DNA actually exists, how much copy number of the DNA. So you can imagine if there's more intensity, there's more DNA. And if there's less intensity, there's less DNA. Duplication and deletion. So we wrote a hidden Markov model way back in 2007, which was kind of state-of-the-art AI at the time, to call these hidden states from the sequence of intensities produced from a machine. Not really intended to do this. And we published a paper, big data, right? So 13,000 samples, a configuration of with and without ADHD, common things being common. ADHD is the most common neuropsychiatric disease in the pediatric population. And we found this network, to keep a long story short. So this is a metabotropic glutamate receptor network. Meaning there's 200 odd genes that are connected by protein-protein interactions. And the way we implicated mGluR, which is a network that you can go and drug eventually, which is exactly what we did, is we looked at the deletions and duplications in this network of genes that surrounded glutamate receptors. So there's 1, 2, 3. There's 8 glutamate receptors. There's a family of 8 glutamate receptors, GRMs. And we were just struck by how significant that family was on its own. And when we considered the entire network, i.e. the genes that interact with it from yeast to hybrid screens, so a different source of a different level of evidence of function, and we paint red for deletions and green for duplications, but there's 2 levels of colors on the charts. There's light and there's dark. So here's Here's dark green, and here's, green is a bad color, I can't find any light green, but here's light red, and here's dark red, right? So the light means this gene is enriched in controls, and the dark means this deletion or duplication is enriching. Just look how dark the picture is all I'm trying to convey. And that's a 10 to the minus 10 significance of it being this dark. So we've implicated this network method. We've developed this network method to find very rare defects. And when you sum them all together over these 200 odd genes that have some protein-protein functional interaction, it's quite significant, 10 to the minus 10. On their own, none of these are significant except for the GRMs themselves. That's what drew the focus to this picture. Does that kind of make sense? So we think mGluR is important, and we made a drug for it. That was ADHD. So the clinicians in this room probably know that autism is a much more difficult phenotype. I think you mentioned how phenotyping is probably more important than large-scale sequence. Here's a good example why. Autism is a diagnosis of exclusion. You can go whole genome sequence everybody in the world, except you're pointing at 200 or 300 different autisms, and it's not very useful. You keep throwing more noise at a noisy phenotype. It's just not very useful. So we thought, ADHD, autism, bipolar, schizophrenia, different labels for the same kinds of problems in your brain. And we thought mGluR would be important in autism. And with more effort and analysis, it actually is. I mean, notice there's more light on this. It's the same network, I guess, plotted a little bit different, but at least I could find light green. Here's a light green, right, on this chart. So it's just a little bit more diverse because I think the phenotype is just much more difficult to capture. If you look at the numbers, we use more numbers. There's 19,000 samples to get to this level of significance, right? So just some insight. And we found a drug. So this implicated the mGluR network. It's in the brain. It's a metabotropics glutamate receptor. You put that into Ingenuity, this drug comes out. A safe drug was under development in Japan, I want to say, 20, 25 years ago for Alzheimer's disease. It passed phase 1, it passed phase 1, which means it's safe, but it failed in phase 2B, which is efficacy. So we thought they had the wrong indication. When we do this kind of analysis on our Alzheimer's data from the adult hospital at Penn, it just doesn't hold. The same kind of signal is not there. So we thought they had the wrong indication. We started a company, bought the rights to the drug, set up a clinical trial, standard type of deal, 9 weeks, dose escalation, 30 kids after washout over the summer, right? So we washed out their Ritalin. And again, this is a phase 1 trial for efficacy, but you can ask questions about how well it responds. And both the clinicians, which is here over the weeks of the trial, and the parents repeat a significant improvement in their child's ADHD symptoms, right? So that's just an example of how big data, relatively rare. This is about 10% of kids with ADHD we're targeting with this defect, but it works in those 10% of kids. So we've all seen some version of this slide. At some point, at least I have many different versions of this slide. But the average clinical trial 10 years ago was, $1 billion over 10 years? So that the cost has doubled or tripled today, and probably the time has not, it has not certainly decreased. And the reason is because the way drug companies work is they just care if the drug is safe. It's hardly as much as if it's effective as if it doesn't kill people. Tylenol is a great example, a very safe drug. Kind of works, not necessarily all the time. All we're saying is we want to inform the decision of how to build out these clinical trials to begin with. So we're in a massive pediatric children's hospital. We've already consented everybody. We know what their genetics are, and we know who said we could recontact them. So for $1 million, literally, a few $1 million, we were able to set up this clinical trial. The company was purchased 2 years ago, so within 5 years, we did discovery, went through the FDA, got through phase 1, and got acquired, only because of genetics and the way we sort of engineered the health system to help us. Right, to take, instead of enrolling 100 people to show efficacy on 10, we enroll 15 or whatever these numbers are. So it's much cheaper, much more efficient, and the health system sort of takes the bulk of the recruitment and enrollment, which is the bulk of the cost of a clinical trial. There's a different way to kind of think about personalized medicine and a way to engineer it almost. I came up with this pipeline of innovation, let's say, just for lack of a better word, but always powered by some kind of big data. So here's a biobank at the Center for Applied Genomics. Right, we do some kind of analysis, we look for a screen, we find risk factors, we use pathways because genes are too rare on their own, and we come up with clinical therapies and rapid proof of concept clinical trials. So that's like a recurring theme you're going to see in my talk today. So any questions on the first part? But that's like a straight genetics, personalized medicine talk. It's going to change. Okay, so let me tell you about another disease. So I'm from Trinidad. The most impactful viral disease in the world is dengue virus. Right? It kills 20. The World Health Organization knows this, and it's been increasing since whenever this is, 1950s. Half the world is at risk, basically anybody living in the tropical belt. About 400 million people get bit by mosquitoes and get infected. Most of them don't even know, about a quarter. About 100 million manifest clinically, some kind of fever or rash or something. 2 million people die 2 million people get very severe symptoms, and 21,000 or so die every year. So 21,000 people die from dengue fever every year. So you guys remember Ebola? Last year it made a big deal, or 2 years ago. 9,000 people died on the worst year for Ebola. Every year, 21,000 people. Ebola at its best year is like 800, usually. Right? So it's a huge problem. We don't actually know why. All kinds of genetics behind it. We want to predict of the 100 million people that manifest clinically, the 2 million or so, the 2% of cases that get sick and go on to die and get shock and hemorrhage. How are we going to do that? Mind you, dengue is a neglected tropical disease. The NIH is not funding this work. So just to give you some clinical background, so the typical, you get bit by the mosquito on day 0. You have a pretty nondescript viral prodrome, right? So you have evidence of viremia in your blood or protein. You have a headache, fever, rash. Big deal. Every virus just about gives you that. But here's where the subpopulation that gets sick gets into trouble. They get shock, they get thrombocytopenia, so their blood changes, they get bruising and petechiae on their skin. And for the longest time, this is how we thought dengue was. This is what the World Health Organization defined dengue as, until they realized it wasn't working. So in 1997, the specificity for dengue was 36%, meaning people with the flu were being called dengue, people with chikungunya were being called dengue, all kinds of non-lethal things were being called dengue. They changed the definition. Now listen to that. They changed what dengue is and how the public health surveillance of dengue in 2009, and that raised the specificity from 36 to 57%. And they said, all right, there's regular dengue and then there's severe dengue. Severe dengue means any one or combination of these. And there's AST and ALT and all kinds of different, not that important. They changed the definition of what dengue is. So how are we going to do this? It's a messy phenotype, very messy, right? Not funded, massive impact. How are we actually going to come up with biomarkers to predict who's going to get sick? Well, we're going to use the public data, and this is Atul's inspiration. There's over 2 million samples of public functional genomics experiments in the public data today, digital samples of experiments over the last 20 years. So high throughput genomics started in about the year 2000, just before the genome was sequenced. And you can kind of see the evolution of technology here. So here's microarray profiling, right? Affirmetrics, this was, the bulk of functional genomics in the 2000s. And here's where NGS started up, Celexa and Illumina kind of started here. And obviously this is going to take over, this technology is going to take over microarrays. But for now, just take my word for it, microarrays are the bulk of the public data. So I did a study on public data from this gene expression omnibus for dengue. And I had a very specific question. I wanted to know people where the blood was taken before they actually got sick. And I wanted to know what happened 30 days out when they actually got sick or fever or hemorrhage or whatever. I wanted to know the outcome 30 days out. But I wanted the blood to be collected before they actually got sick, when it was just a nondescript virus. Again, dengue is worldwide. These are the countries. Nobody's paying me to go to these countries to collect this data, but it's right here in the public data. We just need to look. This is about the data store, the numbers I use, right? So people that were uncomplicated, I compared to people that got severe dengue. And that's a combination, either or, of these phenotypes, hemorrhagic fever or severe shock. Not big numbers, but it came from a big data store, right? And to keep a long story short, top 10 genes from this functional analysis, this is levels of genes, not sequence, is TNF-alpha. TNF-alpha is a known defective pathway in those that succumb to dengue, and dengue pathogenesis. And here's an example of a biomarker I can go test in any one of these countries, right? So each of these is a different country that I showed on the other slide. And this is the log2 fold change of this gene, which gene is this? This is this one, elaine, neutrophil elastase. And just in every country, this thing is elevated significantly. Anywhere from 2 to the 1.5, that's like what, like a threefold up, anywhere from a threefold change to much less. We don't know what the actual value is, but suffice it to say, I would pick this and genes that look like this to go find biomarkers for people that get sick from dengue. And if you can find a signature for dengue, meaning a list of genes, so here's one example of a gene, anywhere in the world you're going to pick it, it's going to tell you if you're going to succumb or at least progress to more severe dengue. If you line up the top 100 of those genes, so to speak, both up and down regulated, we have large libraries of response to drugs that could potentially correct those defects, right? So here's a functional signature for dengue. Let's say 100 genes from most downregulated to most upregulated. We can find signatures of drugs that correct that. So just stretch your mind a little bit. You'll see that here's the green, and these drugs are mostly red. Here's the red. These drugs are mostly green. And there's a gradient in between. And I draw your attention to just very basic questions public data and free data can ask you. So here's estriol. This is estrogen, right? This is a known hormone. Half the population, adult population, takes this for birth control. So the question is dengue or severe dengue reduced in such a population? The main problem is the countries that this exists, they don't really have good public records. We can't really answer those questions. But suffice it to say, we can come up with all kinds of hypotheses to test just from using public data and a little bit of effort. So here's my pipeline of innovation, again, not powered by a big hospital, but by open data that the NIH and other funding agencies have forced us to collect over the last 20 years, right? Meta-analysis is the analysis we use for this. We find signatures of disease. We can find biomarkers. We can line them up to other databases to find drugs. Quick proof of concept clinical trials on the cheap. Any questions on dengue? We'll change directions again. So, like I mentioned, there's one database we talked about so far, this LINCS, Library of Interconnected Cellular Signals. There's a bunch more, right? GEO we talked about. Here's where a public database of functional gene expression. And you have databases that are accessible to varying degrees of openness along the entire chain of translational research. My colors on the slide changed. It used to be more contrast. But at the basic research level, Esteban can tell us about the database of genomes and phenotypes. It's massive open stores of data of literal sequence. There's a cell line atlas. There's ENCODE, which is very deep ChIP-seq and functional analysis of the genome. GEO is functional things, string is protein, UniPro is protein, array express is a variety of things. There's chemicals you could look at if you want to get to drugs. So PubChem is a database of functional side chains on drugs. Ingenuity is pathways. And at the patient level, there's raw EMR and EPIC, right? There's clinicaltrials.gov to test drugs. All of these things are large. All of these things are mineable. And the real power, I think, of translational genomics is integrating all of these across our private patient stores. But there's a big problem. So the data is complex. We really haven't come up with good ways for computers to parse this data and make it machine readable. And I'll give you an example using dengue. So what I thought is, well, we have lots of people that are interested in this. I mean, dengue is one of the most impactful diseases. Why don't we get people to help us? So this is what I had to do for dengue. Remember, this is a disease they changed the definition of, right? Some people called what we were interested in DSS. Some people called it uncomplicated dengue. One guy called it WHO stage 3 and WHO stage 4. Like, who knows what that means? Like seriously. So I mean, it's not hard. No computer is going to know what WHO stage 1 and WHO stage 2 is. But imagine you can go read a paper and see who changed the definition of dengue and very easily figure out what this means, right? So I was like, we should just build a website to do that. And that's exactly what we did. So I pretty much took GEO, which is an open database, been around for 20 years, open FTP server, and built a front, literally, around it so that anybody could go in there, expose dengue. So is this dengue? Yeah. Acute dengue infection. Here's a study, and they just need to go figure out the phenotyping, exactly what you said. The phenotyping of this open data is what's the problem. And very easily we can do that. Very easily I could come to results online because now we have the annotations of who has what disease, so to speak. We can easily compute the back end of probes and match them to genes. Here are the two genes I talked about, Elaine, this is the one I showed you a picture of, on the web with a URL. And once you can put someone on the web with a URL, then you can ask your friends and people on the internet to help you. Right? So what was this? Like 2 years ago, I sent a tweet. Google MD PhD, Genomic, found this guy in Chicago. He was now starting as a first year. Him and his 10 friends just went through and annotated as much phenotypes as they could on my website, right? So we made 500,000 annotations at the time. This number is now 2 million, right? 2 years later. What does it mean? So we paid out 10 grand. This is kind of how we did it. We paid out 10 grand to get him to do this. We asked him to do it twice, meaning The green were samples people annotated twice, meaning if you agreed who had dengue hemorrhagic fever and your friend agreed who had dengue hemorrhagic fever, both of you got paid. If neither of you agreed, well, then it's wrong. I only paid for precision, reimbursed precision. And you can see very high inter-rater. Look how high the green is. So it's more than a 90% kappa inter-rater reliability. So we have good signal, and we've measured it. This entire thing is green now. We're at 2 million. We spent like 50 grand so far. I probably should have showed a better figure. And to kind of prove that there is real biology behind here, we took a look at TCGA, the Cancer Genome Atlas. This was on that public data slide I showed with integration. And TCGA is probably the best funded massive data set of cancer, right? Analyzed over a variety of different technologies. And we asked very simply, what does a gene signature for breast cancer look like from TCGA versus one from the public data? And of the 30,000 genes we plotted, very significant R squared for the effect size of breast cancer case versus control using our database, using TCGA, except ours cost $10,000 to get, probably a lot less for TCGA. Probably a lot less for breast cancer. TCGA costs hundreds of millions of dollars to create, right? So just to kind of drive home the point of the top, and people just care about the top genes for any disease, right? The top 98 out of 200 genes are both overexpressed in both groups. The top 92 out of 200 genes are both underexpressed in both groups. So clearly we recapitulate similar signals to TCGA using an open data approach. And the argument is we can scale it to as deep into breast cancer or any phenotype as you want, because all the data is there. And if you could do one disease, if you ever wonder what a tree of disease looks like, right, you might be a doctor wondering if I should give my patient some drug because it may have symptoms shared to it. Well, this is what a functional representation of disease looks like on a very large scale. I forget exactly how many, it looks like apparently more than 10,000 samples we annotated with a crowd to kind of get to this. We don't need to belabor the point, running out of time. One more thing, so I'm into digital health. So the evolution sort of, to me, of this big data analysis is not really humans doing the analysis, but computers doing the analysis. So digital health is supposed to be the convergence of the digital and genomic health revolutions. And it's supposed to be a way, kind of like lifestyle. We can use digital health to kind of help improve our lifestyle and kind of better embrace precision medicine, so to speak. I don't need to tell this crowd, but there's all kinds of sensors coming out these days. But let me just leave you with this. So the first time I paid attention to AI in medicine, it's just AI period, was probably 1997 when IBM's Deep Blue beat the world Russian chess champion, right? Let's fast forward to today. This is a little dated. Google beat the second best Go player in the world a couple months ago. Just last week, they beat the best, like beat the pants off of him. Right? Go is a more advanced version of chess, apparently, a Chinese version. I don't know. A couple months ago, Tesla showed that computers driving cars are 40% safer than humans. And it's funny how they showed it. There was a crash in Florida, a semi-jackknife, and the car went under the semi, decapitated the guy was gone. And the DOT basically said any human would have done the same thing, and vindicated Tesla and said their cars are 40% safer. So it's very interesting how this is turning out. Well, the more recent stuff, deep learning is the secret, right? So what is deep learning? So deep learning is not new. It's been around from about the 80s, right? It's basically artificial neural networks. And theoretically it's been around, but the reason it's emerging today is because GPU computation, or highly parallelized computation, exists today from groups like NVIDIA, and Google and everybody now is building some kind of AI infrastructure. But more importantly, the big data to kind of train these models exists today. So here's an example of 7 different classes of objects, let's say pictures. Here's what the pixel level representation used in traditional machine learning looks like, not very discernible, but here's what it looks like if you use deep learning. So, and the way this works is, let's think of a line. Most of statistics is based on the equation of a line, y equal mx plus c. With 2 points, I could plot the slope of the line and the y-intercept of the line, right? 2 points, 2 parameters, not a big deal. You don't need big data to work with lines. But some of these models, like the models that predict our weather, there are more parameters in the models than there are atoms in the universe. Like, there's no way to train such models to completion, period, right? So the point is you need a lot of data to get the precision to train these models. And that's why deep learning is making a splash today, because we have enough data, we have the computation, and we have the models. And This is vastly different to lines and statistics. We don't actually know how these things work. Google sat and said, okay, so anybody that went to medical school probably knows visual cognition. There's an entire system. There's V1, there's V2. Light hits the retina cells. It goes to the lateral geniculate nucleus and goes through all these nested hierarchies of cognition to tell you this picture is a picture of a lady. Google built exactly the same thing in a computational paradigm, right? A nested hierarchical network of knowledge and knowledge inference. We don't need to get into the details. And people started to pay attention when deep learning won this competition in 2012, right? So this competition is, you have, since 2010, the goal is to classify 1 million objects into 1,000 categories. And these are things like dogs and cats and bridges, and very mundane things. And the way traditional computer vision works is you need to know what a dog is, or at least know how to define a dog in computational terms, and define a bridge in computational terms for all these blue points. For these green points, we just tell it it's a dog. So a human tells us it's a dog, the computer figures out how to define it. It figures out if it needs a curve, if it needs contrast, if it needs colors. Deep learning learns the features automatically, which makes it quite robust and scalable, so to speak. So now, in 2015, when they started, the error rates were 70% with traditional machine learning. 2012 significantly improved the error rates, somewhere at close to 80-85% today. Google and Microsoft could beat a human at telling a fuzzy, out-of-resolution picture of a cat is actually a cat. 97 plus 98% accuracy rates. So this is why every other Monday morning, some medicine and deep learning and AI is kind of in the news. So a good example of what we're using this stuff for is breast cancer. There's this problem in breast cancer such that we now have open competitions to try to solve. So here's one competition to try to solve a digital mammography screening problem. Not too important about that slide. What is the problem? The problem is, out of every 1,000 women screened, 100 women are recalled for some problem on the mammogram. And of the 100 women recalled, only 5 of them have a problem. So 95% of the recall is false, so to speak. And that's a huge problem. In fact, some of the studies that have studied mammograms have showed they've really done more harm than good over the 20 years of screening, meaning we are not really, we are reducing the rate of breast cancer, but it could probably be done for a lot cheaper by manual breast exams. There's a huge controversy as to the utility of this kind of mammographic screening. We want to improve that with computers. We want computers to achieve 97% accuracy rates, just like we know they do for dogs and cats and mammograms. Just some more details. There's 4 views per mammogram because there's 2 breasts and 2 views per breast. There's screening, and if you have a problem on screening, you have even more views. So there's all these different input into our system. The outputs, the things that seem to be associated with mammograms or some kind of history, right? The actual history of the breast, if you have implants, if you had previous cancer, your demographics, obviously, your race. So all of this is information we have on the patients to kind of help learn from the pictures what is the diagnosis, right? And here's our first attempt using that same model that won, right? AlexNet in 2012, the model that won ImageNet. We took breast mammograms and pumped it through the exact model. That model was trained on cats and dogs, right? And we pushed our mammographic x-rays through it. The red is mammograms with cancer, and the blue is mammograms without cancer. You can already see separation. This is called transfer learning. We've transferred the learning that a first-year resident has, basically knowing how to discriminate cats and dogs. They could probably go look at some of these things and see some separation. The computer's doing the same thing. Now the idea is to take that computer through residency and teach them how to fine-tune and really pick out microcalcifications and very, very subtle features, which is essentially what residency is for people, and fellowship, right? So here we go, all the red are kind of clustered, and we can see why they're not clustering. So here's a misclustered one, and the reason it's misclustering because it's edge. There's an edge artifact on the mammographic screen, and this is what the computer is picking out as a signal. So we need to fix these kind of problems, and the way we fix it is with more data. This was only 500 mammos, right, that we looked at. To keep a long story short, Michael is an SFSU master's graduate now. Graduated last week, and that's how I got invited to this talk, so thank you, Michael. But his work was to fine-tune our data at UCSF, basically to try to win the dream, right? So we took our data at UCSF, we trained our own model, and we submitted it to the dream, and the area under the curve is 0.75, which is pretty good, which probably is better than a human. We haven't tested it and validated it, as yet. But it's pretty good, and it's an example of how we use routine data from UCSF, and we can apply it to somebody else's data. right, in Seattle, so that's where dream data comes from, and have some evidence. This is very small data relatively. We're scaling this up to about 700,000 pictures of breasts we have now for mammograms, and that's quite a process. And again, just to kind of give you some complexity, the way that AlexNet, that model that allowed us to plot this picture worked, the way that worked is trained on an ontology of things. So dogs and cats are animals, right? So there's some knowledge tree that the pictures are labeled with, and that tree is used to train the models, to basically recapitulate the tree or discriminate the tree or predict an incoming picture where it would fall in the tree. The tree is used in training the model. So we are trying to do the exact same thing for radiology, right? So we want to train models, not pre-trained on dogs and cats and bridges, but on x-rays of feet and hands and backs and things that are of use, because I can't imagine knowing anything about a dog or a cat is that helpful. Well, we'll find out, but it just seems that we can do much better with bigger models, bigger data. This guy, Jay, is a first-year radiology resident at UCSF. He won Stanford's biohackathon with this idea, so we're working to implement this, and this is about 10 million This is about 10 million radiographs we're working with to build this kind of baseline model on which to train mammograms, on which to train, NG tubes, whatever you want. You sort of need this baseline model, and this is kind of our aspirations. So again, we have our pipeline of innovation here, except pictures and images and pixels are the real data points that we are analyzing. Deep learning is the magic that makes it happen. Suffice it to say, we could very easily test how much we improve the recall rate for breast cancer with this kind of report. The data is already there. We have 700,000 mammograms we're looking at. And just to kind of tease you, it works for breast cancer. It also works for skin cancer, right? Deep learning, digital health, I think is the future. If you could put deep learning into a phone and look at your mole and say, is this melanoma or not? I think that's quite possible, and that is definitely the future. With that, I just want to quit and end, hopefully. I probably didn't go over time, but I think data is power, data is revolution, and data is frozen knowledge. So Atul said that, I'm not sure when, in a TED Talk somewhere, and I completely endorse it. Thank you very much.